Hello, Bio30, and welcome to our lesson on DNA. Today we are going to look at how DNA and its counterpart RNA are structured, as well as we're going to focus on who was part of the discovery of DNA and how that became a really important part of the understanding of how inheritance occurs. So by the end of today, your goal is to be able to summarize historical events that led to the discovery of DNA. So some key people we're going to talk about. You need to be able to describe your structures of DNA, meaning you need to know all four bases, what makes up the backbone of DNA, which we call the rungs, the backbone, sorry, and then the rungs are the bases. We're going to also look at explaining the different types of DNA. So really quickly, I just want to remind you of your favorite lessons from Bio20, which is macromolecules. Back in macromolecules, we talk about amino acids and nucleotides. Amino acids are proteins and nucleotides are nucleic acids. Now, what we're going to focus on for this chapter is how does the nucleic acids or the nucleotides actually help design proteins? That's the whole point of this chapter. So there's actually a big connection there, but we don't actually talk about nucleotides or nucleic acids in Bio20. We focus mostly on proteins. So what are nucleic acids? They are the biological chemicals that direct the growth and development of every organism. So every nucleic acid is made out of 3 or 4 letters, A, T, C, and G. So that's DNA. DNA is made of A, T, C's, and G's. So we're going to look at 2 types. Now, you may notice it says and you, and we're going to get to that in a second. But DNA is deoxyribonucleic acid, and that's what's inside our cells. So you will notice that there is a lot of things it does. It's for hereditary, meaning it's what we pass on to children. It's inside your nucleus. It makes up your genes, which are the things that make you you. So your hair color, your eye color, for example. It's condensed down into stuff called chromatin and chromosomes, which we'll learn in our vocab, as well as it's double-stranded. And like I said, it uses the bases A, T, C's, and G's. Now, we're going to talk a lot about how ATCs and Gs are mixed and paired. Now, you'll notice that there was also and U. So it says up top, they're made of nucleotide monomers A, T, C, G, and U. Well, U is called uracil, and uracil is actually only used in RNA. And RNA is ribonucleic acid. So there's a difference in the way the structure of the sugar is made. So DNA and RNA look a little different because of their sugars. So ribonucleic acid makes up a bunch of different types of RNA. There's 3 types, and we're going to talk about 2, and well, we're going to talk about 3, but we're going to focus on 2 of them in this unit. DNA actually helps build RNA, so we're going to get to that as well. And mRNA specifically makes the proteins in your body. And the reason why we need RNA is because it can actually leave the nucleus where your DNA cannot. So we build proteins in ribosomes, which are outside of the nucleus, so we need it to be able to leave. RNA is unique in the sense that it's single-stranded instead of double-stranded, and we replace thymine, or T, in the DNA with uracil, which is U. So this is what it looks like. So you'll notice that RNA here, okay, this is RNA over here. We call this a ribose sugar. And over here, we call this a deoxyribose sugar because you'll notice on this sugar here, it has an oxygen, but on this sugar here, you're missing the oxygen. So D, removing of the oxygen on the ribose sugar. So that's how DNA kind of got its name. We're going to focus in on who made some pretty key discoveries when it came to DNA. Now, this is a lot of information for Hershey and Chase, but really what I want you to understand is just that in 1952, they really discovered that DNA was the transforming principle that carried genetic information, which just really means that DNA is passed from one individual to another. So they did this entire massive experiment that used bacteriophages, which are essentially a virus that infects bacteria. And what they did is they made one that had radioactive DNA and one that did not make radioactive DNA. And then they allowed those bacteriophages to infect cells. And then their DNA would blend into the DNA of those infected cells. And then they would look for that radioactive DNA. And it turns out that that's what happened is the radioactive DNA blended into the DNA of that of that bacteria, and when that bacteria split, that DNA was passed from the original bacteria to a new bacteria. So essentially, they were like, hey, we think DNA is passed from one individual to another. Around that time, we also had Rosalind Franklin, who in 1952 also produced the very first photograph without realizing she did at first, but produced the first photograph that revealed like an X-shaped pattern of DNA, which was allowing Watson and Crick in 1953 to build a model that demonstrated that DNA was a double helix shape. Up until that point, nobody knew that it was a double helix shape or how DNA actually looked, and people overcomplicated it, and nobody could actually define the structure of it. Now, Rosalind's picture was super, super important because if it had not happened, then Watson and Crick probably the shape of DNA is. 1990s to early 2000s, there was a huge project also that occurred during that time in which we called the Human Genome Project. So it essentially mapped all the genes in your body. And we're going to talk about gene mapping and how we know what's on each chromosome later on throughout this chapter. So we're going to look at we're going to look at the different types of DNA. So DNA comes in a variety of different types. We're only going to focus on in this chapter nuclear DNA, stuff that's found inside your nucleus. That being said, it's very cool that mitochondria, which are found inside our cells, in all of our cells, that's where we do cellular respiration, has its own type of DNA. And then the chloroplast, which is found inside of plants, also has its own DNA. Okay? So mitochondria and chloroplasts have similarities with bacteria. They're double membraned and they each contain their own DNA. So there is a lot of different types of theories of how the first cell got mitochondria into it and why do we have mitochondria today in our cells that help us. But the one thing that they do know for sure is that mitochondrial DNA is actually passed from mother to children. So mitochondrial DNA or mtDNA is essentially traceable through the mother's line. So if you are a female and you give birth to a female, and then that female gives birth to another female, you can trace your lineage through the mitochondrial DNA. This evolutionary history and lineage is actually called the mitochondrial Eve, and we're going to watch videos later showing you and discussing about what that means. Now, one more thing we have to kind of focus in on is what is inside DNA. So DNA itself has sections to it called genes, and these genes code for certain characteristics. So all of your DNA has sections on it, and then it'll be like, this is going to be the section that tells you your hair color, your eye color, your height. And every single one of those genes is made up of DNA that has A's, T's, C's, and G's. Now, your DNA is long, stringy. It kind of looks like spaghetti inside your cell. It's a long thing called chromatin. But in order for DNA to actually fit inside your nucleus, it has to condense down. And when it does that, it can condense further down in replication, which is a process we'll talk about later. And what it does is it creates these X's. So if you've ever seen a chromosome, it'll be in an X shape. Humans have 46 chromosomes, but they're paired up. So we really say that humans have 23 pairs of chromosomes in their cells. What we're going to focus on throughout this chapter is how are those chromosomes replicated? How do we read those chromosomes? What happens when there's a mutation and all of those things. But it all starts with DNA. So this is just kind of a transformation showing that here's your DNA. And if we look at how DNA is condensed down, this is a chromosome right here. That chromosome will be found inside the nucleus, which is found inside the cell. So you could go big to small or small to big, and you'll see that this section on the DNA is a gene. So you would then condense it down. So somewhere on this chromosome is this gene, which is found inside the nucleus, which is found inside the cell. Now, if we take a closer look at DNA as a whole, you're going to see that DNA, like we said, double helix, has adenine, thymine, cytosine, and guanine are 4 base pairs. Each of them are held together by hydrogen bonds right in the center. We're going to focus on that in a tiny bit. And then all of these backbones, this blue ribbon is actually made out of phosphates and sugars, and they're held together by covalent bonds. So when I talk about A, T's, C's, and G's, I'm talking about the nitrogen base. But when we talk about the entire part of DNA being the nitrogen base, the sugar, and the phosphate group, we're actually referring to something called a nucleotide. A nucleotide has those three things in it in order to be referenced as a nucleotide. So you need to know that a nucleotide is a nitrogen base, a sugar, and a phosphate. It's also set up in a very specific pattern. So if you look at this picture here, this is what it would look like. Your phosphate would be attached to one of your parts of your sugar, and then the internal part of that sugar has a nitrogen base attached. Okay. Now, as a reminder, we would then look at these based off of having a an oxygen or not. So this one over here is showing a lack of oxygen. So this is part of the sugar for DNA. And this is showing an oxygen. So this is part of the sugar for RNA. So if you look again, here's the chemical structures of what those look like. You won't be expected to do the chemical structures, but you will be expected to understand the groupings and the pairings. So purines and pyrimidines. Purines are much larger, and they are adenine and guanine. So adenine and guanine are two of our nitrogen bases. They fall in the category of purines. They have two ring structures. Where pyrimidines, cytosine and thymine for DNA, okay, are single structures, and then you have uracil that's part of RNA. Now you have to understand that when it comes to these lovely things, they pair up in a very specific pattern. And that's called Chargaff's base pairing rules. So we're going to talk about the pairing rules. So thymine and adenine always pair together. You can see that here, thymine, adenine. The reason they always pair together is because they can make two hydrogen bonds based off of the structures we have. Where guanine and cytosine make three hydrogen bonds based off of their structures. So we say thymine and adenine always pair together. Guanine and cytosine always pair together, okay? We call this complementary base pairing because they're not identical. So adenine and adenine don't pair, thymine and thymine don't pair. That'll never happen. We call them complementary because they match each other's shapes. Because of this, that means your DNA, if it has 10% adenine in it, it will have to have 10% thymine. So this is called the Chargaff's rules, which essentially just means you're going to always have an equal amount of guanine to cytosine and thymine to adenine. So this is just an example, okay? If you look at this and we look at what's happening up here, okay? It shows that in this particular human, 29 of your base pairs were adenine. So that means 29 of your base pairs have to be thymine. If 21 of your base pairs is guanine, then 21 of your base pairs has to be cytosine. So if we were to go through, and do these questions, you would then do the same thing. If there's 31% of adenine here, you would have 31% of thymine, which means how much of your DNA is left over. So 62% of your DNA is taken up by adenine and thymine. So you would have to take 100% of your DNA, subtract your 62, and that's where you would get your leftover DNA. Okay, so that would be what is your cytosine and guanine. So there's a bunch of questions in your practice that I would like you to stop and pause the video and try. You can work with a partner quickly if you want, but essentially it's just getting you to make some new DNA, so showing me the complementary base pairing, as well as talking your way through how do you know how much of each percentage of nitrogen base pairs there are. The last few things we're going to talk about is how is DNA actually set up. So you'll notice that on this side, DNA's sugar is pointing up, but on this side, DNA sugar is pointing down. And that's because DNA is actually anti-parallel, meaning it doesn't run exactly the same way each time. One runs up and down and the other runs down and up. So we talk about how DNA is attached based off of where the phosphate group is. So we call this the 5' end because our phosphate is up at the top attached to the 5' section of the sugar, okay? Where down here we call this the 3' end because of where our OH is attached. Same with over here, you'll notice the other part here is the 3' end because that's where the OH is, and so the 5' is down here. So we're going to talk further about what is the function of DNA in other lessons. But for now, that's kind of just the gist and the general idea of what DNA is. So if you could take a minute to make sure all your notes are in, that you tried those practice questions, You will be kind of halfway done this lesson and you'll move on to cell cycle soon. Have a great day.